starting with Galen, probably the world's first formal anatomist. He lived between 130 and 200 AD and was Roman and felt the stomach to be an animate being. So we had a different perspective in terms of these people thought that in fact the stomach was what kind of drove our behaviors. And what's interesting about that is that he may not have been too far off when you think of some of the gut hormones like leptin and ghrelin that are really driving our satiety perceptions and our appetite. There is a certain component where there are gut-derived hormones that are probably influencing our perception of food. The stomach rouses the animal, searches it to go in search of food, and some of the actual terms that we use were coined by Galen. I guess several thousand years ago. So intus from intestines means within, so it was a container, and stoma is a mouth or an opening from stomach. Pancreas means pan, all-encompassing, and creas is like a meat or a tissue. So moving on here, we're jumping up here to about the 1100s. So Avicenna was a 11th century Islamic anatomist. One more comment. Let me see if I can go back here to Galen. What's interesting is that he made some great observations in terms of anatomy, but with one small deficiency, and he didn't have the opportunity to look at humans. So many of his observations were made with animals and simians, apes, which actually have some differences in anatomy relative to humans. So again, 11th century, we began to focus a little more on the function of the organs versus the anatomy, and it was not until the Renaissance times, in 1600s, that people started to do more accurate appreciations of the human anatomy. Vesalius is probably the grandfather of human anatomy, published De Humani Corporis, what the fabrications of the human body in 1543. And what he did is he started to expand some new nomenclature because he was actually, in fact, able to examine humans and do anatomical surveys on humans. And he coined the word duodenum, and duodenum is an ancient word that means basically the breadth of 12 fingers. So if I hold my fingers up, you can see if I were to put 12 fingers side to side, that's about 10 inches, which is about the length of the duodenum. That's the first part of the small intestine. There was also a concept of some moralization of the organs as well, and so here's a print made by Leonardo da Vinci. Again, we can start to see that there's more anatomical semblance to really the organs as we know them today. Similarly, associating gut function and behavior, and let me see if I can do this in my best Scottish accent. So Lady Macbeth is waiting in the castle for the soon-to-be King Macbeth to come back from the battle. She's secretly plotting to kill King Duncan, and she says, "Come to my breast and take my milk for gall, you murdering ministers." So she's summoning this gall, this emotion to kind of give her the courage to create to follow through with this heinous crime of convincing her husband to kill the king, which he ultimately does. I'm going to go a little farther up in history. This is the bizarre case of Alexis St. Martin. Mr. St. Martin was a 19-year-old man who lived up in Mackinac Island off of Michigan, and he sustained a gunshot wound to his upper abdomen. Here's the doctor, Dr. William Beaumont, who cared for him, and here you can sort of see the position of the gunshot wound. And what's interesting is that, so this is actually from the writings of his good doctor. Let's see, being at a distance of not more than 1 yard from the muzzle of the gun. Now you can imagine this is probably a 50 caliber gun. I mean, he took a pretty good hit to his belly. And what happened is over the course of the following year, this wound formed a cicatrice. That's a sort of a outdated word for scar. And so this is 1823, scarring over. And by 2 years later, he'd regained his full natural health and strength with one issue. He had a gaping hole from his external body to the inside of his stomach, through which good Dr. Beaumont did what any self-respecting doctor would do, which was conducted experiments. So, so after considerable difficulty and great expense to me, I love this, they succeeded in bringing him from his Canadian home down to Mississippi, where the doctor performed a number of experiments. And some of this is the actual transcripts of the writing. So looked at the stomachs, its secretions have never been so fully offered to anyone. This is true, really. If we're thinking it was just a few hundred years earlier that anatomists were able to just even begin to dissect human specimens. I like this. I submit a body of facts which cannot be invalidated, and their worth will be best determined by the foundation on which they rest, the incontrovertible facts. I love this guy's ego. But anyways, what he found is he made some interesting observations. He took this man, and over a series of several hundred courses of experiments, he tested the secretions in the belly. He would make little, he would tie pieces of food and vegetable matter onto strings and pass it into the hole and kind of observe it and tease it back up and see what happened to it. And he made some interesting observations that really that hold true today. So his book was the Experiments and Observations in Gastric Juice and Physiology of Digestion. That's where this text comes from. And he showed a number of different things. One that sticks with us today is that the process of chymification, as the term as he used, involves hydrochloric acid. And as we'll see later in this discussion, hydrochloric acid is an important component of digestion. And let me just take a moment. Can everyone understand me okay? I'm not speaking too quickly. We're good? Okay, I see a lot of heads nodding. Okay, great. So let's just go through some basic anatomy and function. Too commonly, I think that medicine tends to subdivide our anatomy. We have the otolaryngologist who takes care of the ear, nose, and throat. You've got your gastroenterologist who might take care of you from here to here and, well, the backside. And then you've got your hepatologist who takes care of your liver. And really what I'm going to try to stress to the group tonight is that there's a concept of digestion which really blends together a lot of these different concepts. And all this whole system beautifully works together or fails to work together. So there's a Chinese expression that says, problems here, problems here. And it works the other way around. So problems here, problems here. So could our gut cause allergic problems? Could problems that are involved with our face or mouth or production of saliva affect the remainder of our digestion? I think so. So antiquated paradigm that the gut only serves to digest food and eliminate waste. And I think that by the end of this discussion, I'll have everybody here convinced that the gut's a very dynamic, important, and easily influenced organ. So the small intestine is 20 feet long. If I were to lay it out flat and kind of coil it back and forth, it would cover about 4 square feet. But if we were to open up this intestine and open up all the involutions and the rugae and the infoldings of the gut, it would turn out that the lining of the gut would cover about 3,000 square feet, which is about 300 square meters. And I did a conversion this afternoon to meters because I just want to emphasize the fact that the lining of our gut is 3,000 square feet. That's what our immune system needs to survey. A virus is about 1/billionth of a meter in diameter. So here's our immune system looking for something 1/billionth of a meter with 300 square meters. So I think it's really remarkable what our body's able to do. So this is just a little cartoon that shows how this works, our small intestine. Here are these involutions and crypts. These are formed with what's called a villus, and the blood supply that goes up in the middle is called a lacteal. And this is lined by these single cell layer, they're called epithelial cells. And epithelial cell is like a skin cell, whether it's a epithelium on my arm or epithelium in the gut, epithelium in the vagina or rectum. These are specialized cells that are used to kind of coat surfaces. And these are all covered with these little microvilli. And these are these delicate little hairs that allow our intestines to create these 3,000 square feet of surface area. So what is it that initiates all gut function? And I think it's important to talk about this. This is the vagus nerve. It's the 10th cranial nerve. Most of the cranial nerves that originate in the brainstem serve cranial features. So the ophthalmologic nerve affects the eyes. The glossal nerve affects the tongue, and hopefully mine is working well right now. Facial nerve and a trigeminal nerve are the sensation and muscles in the face. But yet we have our 10th cranial nerve that starts in the brain and innervates our entire abdomen, all of our gastric functions, which I think is pretty remarkable. Just anatomically to think of, most of the cranial nerves are short. They're maybe 6-8 inches, and the vagus nerve has this long extension, and all this is important. So vagus nerve functions, let's start up top here. So production of saliva. Under the correct mental stimulus, our bodies are going to start to produce saliva, much like Pavlov's dogs. So that's an important component in the initiation of our digestive process. The saliva is made of salivary amylase, and 30% of our digestion of starches begins with the process of mastication, chewing, as well as salivary amylase. Gastric acid secretion, I've alluded to this already. If we're not prepared to digest food, if our brain isn't working well, if we're not making the appropriate steps to get into the right frame of mind to eat and digest, we're not going to make appropriate levels of gastric acid. Pancreatic enzymes, this also alludes to the gastric acid secretion. The pancreatic enzymes are secreted as proenzymes. These are inactive enzymes, and they need to be activated by the acidity the stomach. And lastly, the vagus nerve is responsible for the motility and the churning and gurgling of our belly. So which of these two men do you think has the best gut function? Right, this is important. So mouth and teeth, hope no one's offended by the Rolling Stones icon. I mentioned mastication, chewing. So somebody goes into the Sonic burger shop, you're sitting in your car, and you gobble down a Sonic burger in a matter of 10 seconds or so. What kind of information do you think that's presenting to the gastric system, and how are we presenting our foods to the gut? So mastication is important. Smell, the sense of smell, similarly works through the brain and through the vagus nerve to get our digestive processes ready. And I also like to, when I speak to my patients, I think of the image of a young boy running back to the camp with a rabbit that he killed over his shoulder, and that the rabbit was sweating, and there's the smell of the blood, and as he turns the corner towards his caves, he sees the fires are going. This is all part of his digestive process. His brain is actively working to start preparing the salivary enzymes, preparing the gut to do its process. And I talked about saliva already. Stomach, acidification, we talked about. Acidification is important for some of the processes I mentioned. It's also important for deconjugating salts. So magnesium and calcium, we need to have acid to break these salts, break these ions from their salts. And so many people these days are taking these, what are they, the little purple pills, proton pump inhibitors or something like that. We're gobbling them down. You read the package insert, it says to be used for 6 weeks for very specific diagnoses. People are taking them for all this time. Those block the production of acid. So concurrent with that is the fact that osteoporosis, osteopenia, weakness of the bones, is increasing at a dramatic rate in men, whereas it used to be just a condition of the women. Could it be because we're not allowing ourselves to acidify the gut from all these medications? I don't know. Mixing, we talked about. The immune system. As I mentioned, the stomach is filled with acid, and if I were to create a castle that I wanted to protect, if I was the immune system, would you want your moat filled with water, or would you want it filled with acid? I'd probably pick acid. So the acidity of our stomach serves as an initial place for viruses and bacteria, pathogens to basically be dissolved. And the acid in the stomach is pretty strong. If the range of all acids and bases goes from 1 to 14, with 7 being neutral, the stomach acid has a pH between 1 and 2, which if I could get a glass of pH 1 and 2, I could probably drop a penny in it and it would dissolve. So it's a very, very dense acidity. And then the production of intrinsic factor. Intrinsic factor is an important cofactor for the absorption of vitamin B12. Moving downstream to the liver and gallbladder, the gallbladder is responsible for the emulsion of fats. Think if we eat a fatty meal and we drink a glass of water, what happens if we pour a tablespoon of oil on top of a glass of water? It floats. So our body has figured out a way to develop a soap, basically make an emulsion with bile that creates a saponification and allows those fats to be mixed into the more liquid, more aqueous solutions as they go down through the gut. The liver is responsible for detoxification and protein synthesis as well. And these are, it's important, it makes clotting proteins and other proteins as well. Pancreas, I love this, give me some sugar. It makes digestive enzymes. As I mentioned, they need to be activated, insulin, and it joins the bile duct at the common bile duct. And lastly, small intestines, 20 inches in length. Here's that 10-inch long duodenum, 12 finger breadths wide. The jejunum is the second part of the gut and is most richly supplied with blood vessels. It's most intimately associated with the majority of digestion. And the ileum is the third part of the intestine. The terminal ileum, right before the colon, is where vitamin B12 is absorbed. And lastly, colon and rectum. The colon does still get some short shrift as a storage organ, but that's really what it does. It stores our stool, but it's very important for the development or for maintenance of our water balances as well, and of course for elimination. Can you folks see over here? Am I moving around enough that you can look by me? All right. So it appears, Hopkins, that your gut feel was only indigestion. So let's see, moving on here. So these are some clinical signs of a poorly functioning gut. We all know these. These are the allopathic. Anyone can see that the gut's not working well. You get a belly pain. This is an inflamed gut. This person is not pregnant. The cardinal signs of inflammation, number 4, there's calor, rubor, tumor, and dolor. These are Latin words. Calor is heat and warmth. So just imagine if you cut yourself, how your body gets warm. There's dolor, which is the Latin root for pain, dolor. It's painful. There's tumor, there's a growth that develops. And lastly, there's rubor, which is redness in a cut. So, you know, what we're seeing all those 4 cardinal signs of inflammation in this poor person's gut. Other signs of a poorly functioning gut. All right, moving on. And of course, constipation and diarrhea. Now, these are easy clinical signs, but I want to talk a little bit about functional signs of a poorly functioning gut, because this is when we really start to expand our thinking and say, how is it that our gut function, our immunity, is starting to influence the way that our whole body works? So, neurological, behavioral, and psychological. I'll look over a couple studies later on in this talk that demonstrate that. Skin changes and rashes. Everyone's familiar with celiac disease, right? There's a finding in celiac disease which is pathognomonic, and that long medical word means that if you have this finding, it's diagnostic of the disease. So if somebody has the pathognomonic skin finding of herpetiformis dermatitis, it's a distinct finding, that means without question you have celiac disease. So why is it that all the dermatologists that take care of skin conditions can't seem to think that maybe that part of the etiology, part of the cause could come from the gut? So as we start to expand our thinking and taking care of people, we say maybe that chronic rash, maybe those chronic hives, maybe that eczema or the psoriasis might be due to something going on in the gut. Nutritional deficiencies goes without saying. Autoimmune conditions, we'll discuss again celiac disease a little later this evening, and I think my personal belief is that the future is going to hold that a good number of autoimmune conditions are born out in dysfunction that comes from the gut. And lastly, inflammation and pain, I already mentioned that. So let's talk a little bit about the microbiome. I just put this slide up because I love this picture. This is an article by Michael Pollan, who writing in the New York Times Magazine a couple of years ago, and he referenced some of the papers that I'll go over tonight. So microbiome by definition is the sum total of the microbiological species in any environmental system, and the environmental system that we're talking about naturally is the gut, but this could be a fish tank, or it could be a lake, or it could be a forest. What we see up here is these are bacteria up on top. The second slide, I'll get it out of people's way. These are fungi, these are yeast species, and we can see these are mycelia, like what mushrooms grow out of. And here we have sort of these branching yeast species. And these are protozoa, and more specifically, they are parasites. And this is a protozoa which is pretty commonly found in some of the gut testing that I do. It's called Entamoeba histolytica. And here we have it has ingested some human red blood cells. So probably don't want those in us. Put this in for Ed. So microbiome by the numbers, our body's made of about 10 trillion cells. It's thought that we contain something like 100 trillion bacterial species. It turns out that the bacterial load in our gut weighs somewhere between 2 and 3 kilograms, which is somewhere around 5 pounds. If you look at all the genes associated with the human genome, and this has been brought out by the Human Genome Project, which was completed maybe 10 years ago, humans have some 22 or 23,000 genes, but if I were to add up all of the different genetic components of the microbiome in my gut, it might be somewhere between 1 and maybe 9 or 10 million. So understand that these genes of these microbiome, these creatures that live alongside and with us, symbiotically with us, or maybe antagonistically with us, they're metabolically active. So if I eat food, if I eat some vitamins, if I take in minerals, all of these bacteria have their own designs for some of this nutrition. Amino acids, same thing. So by the numbers, 500 species of bacteria in the gut, I said 2 kilograms, and a good amount of our fecal solids are spent bacterial products, almost 100%. So as we go through the gut, we see that this is the concentration of bacteria in different parts of the gut. So this is 10 bacteria per gram of stomach tissue. That's not much. Bacteria are pretty small. As we get down into the duodenum, that first part of the small intestine, we're looking at 1,000 bacteria per gram. Ileum is about 10 million. By the time we get to the colon, that's a 1 trillion microbiological, I guess, bacteria per gram of tissue. So microbiome influences human health. This is from PLOS One in 2010. And I'm sorry this is kind of hard to see, but I'll just read the title. The gut microbiota in human adults with type 2 diabetes differs from non-diabetic adults. And what they found is that in diabetic adults there were lesser number of the Firmicutes and Clostridia phyla. So we're starting to get a sense that the bacterial balance inside of our gut, much the way that Ed Jones alluded to this before my talk, can certainly influence the way we extract energy from our food. And here's a great study. Again, this is published in Nature in 2006. And if anyone's ever read Nature, I mean, this is the most complicated journal I've ever read. I can barely read the titles of these things. But suffice to say that this is a study that looked at mice. And OB, these are mice that are genetically bred to be obese. DIO stands for diet-induced obese mice. So these are normal genetic mice, but they're fed. And with all these mice, there's no significant difference in their chow consumption, initial body fat or weight. And what we find is when we find that with these mice that are destined to be obese, and these are what are called germ-free mice. They're mice that when they're born they have nothing in their guts. So they're like a tabula rasa. The scientists can instill bacteria to try to find out what's going on. And when we instill bacteria from a lean donor, look what happens to this mice. Their increase in body fat is less. Remember, these mice are genetically predisposed to be heavy. And by the same token, dietarily induced obesity, same thing happens. If we give mice the microbiota, the microbiological I guess consortium from a lean donor, again, they don't get as big as the mice that are colonized with bacteria from an obese donor. Another study here, microbiome influences mouse weight. And there was one thing here that I want to say. Oh, here it is. This is the last sentence. Our findings suggest that the gut microbiota is an important environmental factor that affects energy harvest from the diet and energy storage in the host. And this was published in PNAS, another reputable journal. So 99%, we're born 99% human, we die 90% microbial. And I just want to emphasize a little bit how important how many systems are in place for the human mother to begin to make suggestions as to how her child is going to be colonized in the gut. So starting here with the mouth, we see that there are bacterial species that are passed between the mother and the child. So a mom's kissing her baby and touching it, what have you, is starting to inoculate, starting to develop this child's gut. Same thing with the external breast. There are bacteria and yeast species that live on the skin that in the course of nursing are taken into the child's gastrointestinal tract and influence the child's microbiome. The uterus was previously thought to be fully sterile, and now we know that there are some bacterial species that again are in the uterus to hopefully colonize this young child and prepare its gut for life out in its environment. Internal, this vagina, of course, the baby is exposed to some bacterial species while passing through the vagina. And here's an interesting study. If a baby is delivered by a cesarean section, that baby has a microbiota which is more closely approximating the bacterial microbiota of the OR workers and the hospital staff versus a baby which is born vaginally, and that microbiota more closely looks like that of its mother. And this is published in PLOS One, PLOS Biology. Again, this is a really reputable journal. And here's the thing that I find most interesting, is there's some data coming out now to show that there's an active transportation from the gut through our lymph, through our bloodstream, to actually instill certain biological species into mother's breast milk. So these are all systems that are set in place to hopefully develop the baby's healthy microbiota. And another study looking at PLOS One, for people that are healthy versus antibiotic treated, what's affected is the stem cell populations, and stem cells are the cells, they're the progenitor cells for all of our different tissues. So for those babies that are treated with antibiotics early in life, we're seeing that there is long-term physiology versus normal long-term physiology, as opposed to babies that are antibiotic treated. See, the graphic here shows fewer gut microbiota, a less diversified gut microbiota. These babies have long-term physiology. So really our health begins potentially in the womb, and unquestionably as we leave our mother's womb and as we either are breastfed or kissed or what have you. So gut and nutrition, of course, the gut's responsible for absorption of the macronutrients. It's influenced by pathogens, toxins, medicines, cortisol. And these are two photomicrographs. Here we have a normal gut. Again, we see these beautiful villi, these epithelial cells lining the villi. If we had very, very good eyes, we'd see those delicate little hair-like microvilli extending from these cells. And here's a picture of the cell types of someone with celiac disease. So, you can see what the absorptive potential is of this top slide. You can see what's going on here. There's all this inflammation. We used to think that the gut was just absorption of nutrients and then a vessel for toxins, and I think that this image of the gut just being a trash can has kind of gone by the wayside. So we know the gut's important for detoxification and conjugation of toxins. So Again, this could be an hour discussion talking about phase 1, phase 2 detoxification, but phase 1 detoxification is when we take toxins in our body, and based on liver pathways, we make these toxins more reactive. This is a brief intermediate that we quickly want to bind these toxins onto what's called conjugates. And conjugates might be a methyl group, it might be a glucuronide, it might be a sulfur-containing group. But what happens is that when these conjugated toxins make it into our large intestine, there are bacteria that live in our gut that actually serve to deconjugate these toxins. So now we're left with these very reactive intermediates. So there's a recirculation of toxins, and a lot of this just depends on the bacteria that are in the gut, as well as the transit time through the gut. So gut and detoxification, we talked about this a little bit. I tell people to test your transit time. That's easily done. I think a normal transit time through the gut is probably about 24 hours. If you can put a food into your mouth and it passes out in your stool in 12 hours, that's pretty fast. I tell people to just eat a big serving of beets and see how long it takes to pass those beets through your system. I mean, you'll know when it comes out in the toilet. If you're a little squeamish about beets, Ed's got some charcoal that he'd be happy to sell you upstairs. Personally, I'd rather have a big serving of beets than a few tablespoons of charcoal, but I leave that up to you. But again, we're starting to get into the concept of what can we do in the privacy of our own homes to assess the integrity of our gut. And if you eat a big servings of beets and you don't see redness in your stool for 2.5, 3 days afterwards, there's a problem with your transit time. We're going to talk about gut and immunity. So the immune system has two jobs basically. It's to guard barriers. So the main barriers in our body are our skin. Am I doing something here? All right, sorry about that feedback. The gut's job is or the immune system's job is to guard our barriers. So there are really two basic barriers in our body. There's our skin and the lining of our gut. And the immune system only has one job, and that's to respond to molecular signals. So it's important as we go through this discussion that there's only one thing that the immune system responds to, and those are signals, which are molecules. And we'll get into that in a little bit. And then you're done with all the hard science. It turns out that about 70% of our immune system is within 1 centimeter of the lining of the gut. And this is officiated by what's called dendritic cells. And here we have a couple pictures of 2 dendritic cells, a couple really nice photos. And you can see these cells are characterized by these dendrites, these long tree-like, branch-like extensions. And in a slide in a little bit, I'll explain to you what these are for. So the immune system responds to molecular signals, and it basically has 2 courses of action. It can either phagocytize, which means to eat, swallow, engulf, something that it doesn't like, and I just love this photo right here. I mean, look at this cell gobbling down this yellow thing. Or it can attack it, and it can throw a bunch of inflammatory chemicals on the toxin or the pathogen. And I like this picture as well. It looks just kind of like a chihuahua, chasing down this target cell here. So let's talk a little bit about gut and immunity, and this is, I think, one of the most important slides in my whole talk. So if you've made it through this far, hopefully we'll get through this well. So here's this dendritic cell right here. This is the bloodstream and the lymphatics, and this is the inside of the gut up here. And what you can see is this thing is reaching up there through the cells with one of these arms. And what it's looking for is it's looking for a PAMP. This is a pathogen-associated molecular pattern. Remember, the immune system only recognizes molecules, okay? So it's reaching up there into the interior of the gut, and what it does is it grabs something, it takes it into the cell, it packages it up, it puts it on something called an MHC2 platter, which allows it to be identified by the rest of the immune system. And with some chemicals called cytokines, these are these interleukins 2, 1, 6, 10, it sort of instructs the rest of the immune system what to do. So what happens if this dendritic cell reaches up into the lining of the gut and it finds one of these beautiful probiotics that we have over here? What does it do? It passes on the news to the rest of the immune system to these T regulatory cells. And T regulatory cells, through interleukin 2, 10, 12, and 14, basically tells the immune system, we can quiet things down. There's really nothing here that we need to worry about. But what if that dendritic cell reaches up through the lining of these cells and brings something down and processes it? And this is a part of a protein from a Candida yeast species, or maybe it's a toxin, or maybe it's some kind of a poison, or a pesticide, or an herbicide. Conversely, what it does is it activates the T helper cell line. And this T helper cell line starts making inflammatory chemicals called interleukin 1, interleukin 6, and tumor necrosis factor alpha, and it stimulates the Th1 and the Th17 cell lines. And I can tell you that, like there's data coming out now, the Th1 we've known about for some time. Th17 is kind of like the new kid on the block, and we're learning that any number of different cancers, chronic diseases, autoimmune diseases are all associated with Th17 activation. So how do we want our dendritic cells to be influencing our immune system? I want mine to be reaching up there into the lining of the gut, pulling down something and telling my T regulatory cells that all looks good. And this is diagrammed here on another slide. This kind of shows a gut with evidence of leakiness. Here are normal tight junctions holding cells together at the shoulder, and here's an inflamed and leaky gut. We have all these different things that can sneak through and influence what's going on here in our immune system. Toxins, alcohol, hormones, medications, dietary proteins. Maybe part of our digestive problem, which started with our mouth and our chewing, with our salivary amylase, with our gut and our production of hydrochloric acid, with our activation of our pancreatic enzymes. What if we're eating proteins that aren't broken down into the 1, 2, 3, and 4 amino acid chains that are recognized by these cells and actively transported across their lining? What if in fact we're eating proteins or eating foods that aren't well digested, and what's happening is we're left with chains of proteins that don't resemble a hamburger, but they represent some kind of a inflammatory pathogen. This is called molecular mimicry, and this is one of the things that a lot of people are thinking is driving some of the processes that are going on these days. So a quick slide about how one way to diagnose leaky gut. This is an elegant and simple test. It's tested by drinking 2 sugars. One that contains lactulose and mannitol. Lactulose is a big sugar of high molecular weight. It doesn't pass across the lining of the gut. Lactulose or mannitol is a smaller sugar, and quite simply you drink the solution, and 6 hours later you pee into a cup, and if there's lactulose in the urine, how did it get there? It got there across a leaky gut. So what's driving autoimmunity? Here we see from 1950 to 2000, multiple sclerosis, type 1 diabetes, asthma, autism, Crohn's disease, all on the rise. Well, here's a graph right here of genetically modified organisms. Is it possible that with the advent of the leaky gut, with these dendritic cells that are reaching out to present these different components of our environment to the immune system, that genetically modified organisms might be associated with the rise of autoimmune diseases? I just want to comment that these are associations. I can't tell you that this is causative. Okay, what's driving autism now? This isn't known to be an autoimmune disease, but look at the rise of autism over the years. And this red line represents glyphosate. Anyone know what that is? It's Roundup. So something came out just a few months ago where the U.S. government finally declared that Roundup is carcinogenic, and I can assure you that Monsanto's got billions of dollars worth of lawyers that are fighting that in court right now. But what's driving autism? So it could be organic food sales. So I'm just showing you these slides to say that these are associations for us. So my questions are: is this immune nurturing food? It's not the greatest slide on the screen, but I don't think so. It's packaged stuff. And this, I was driving around one time and I saw this sign, and seeing this makes me kind of hungry. How about you guys? I mean, it's like, I can't wait to get out of here and get a bologna and Velveeta biscuit. And I might even ask for mine with double Velveeta. See if they'll give that to me. So we do know that our genetics are influenced by our water. They're influenced by toxins. They're influenced by our air. So our genetics really haven't changed in thousands of years. But we're seeing a rise in autoimmune disease. We're seeing a rise in cancers. We're seeing a rise in autism. Why is that? It's because we're influencing the environment that's interacting with our genetics. Matteo Fasano is a doctor. He's the director of pediatric gastroenterology at a small medical school known as Harvard. And I think this guy deserves a Nobel Prize for medicine, because there's only one autoimmune disease that we know the genetic markers, the environmental trigger, and what's going on in the gut that causes that, and that's celiac disease. So we know that to really be a true celiac, you have to have something called an HLA DQ2 or DQ8. These are genetic markers. Along those same lines, we've known for 100 years that celiacs are sensitive to gluten, and in the presence of gluten, they can develop an autoimmune process. And what Fasano figured out is that there's a protein called zonulin that's in association with kind of those holding the shoulders together of those cells that I showed you. And people so someone can have the genetic predisposition for celiac disease. They can be exposed to gluten, but if they're not spilling zonulin into their bloodstream, they don't have the outward manifestations of celiac disease, which is what we see here. Conversely, on the spectrum, we know that people who may not have the genetic markers but have a leaky gut, as demonstrated by the zonulin or the leaky gut test, and have exposure to gluten, they too can have not celiac disease classically, but they can have celiac symptoms. So the gut and neurology, this is from the New York Times this last June. It's called Gut Feelings. Can the bacteria in your gut explain your mood? There's more and more data coming out that's really associating the bacterial, the microbiome in our gut and how it affects our mood. Here's another study. This is a randomized, double-blind, placebo-controlled pilot study of probiotic for chronic fatigue syndrome. Now, the gold standard of studies in science is a double-blinded, randomized, controlled study. So this one really fits the bill in being the best study that can, we think, that can look at the question. Basically, it says here, we found that a rise in both lactobacillus and bifidobacter in those taking whatever the supplement was. There was a significant decrease in anxiety symptoms amongst those taking the probiotic versus control. So they found that in people taking these probiotics, they had statistically decreased amounts of anxiety. So we're going to get now into gut dysfunction, diagnosis, and treatment. I really wanted to spend some time going through the background, the anatomy, the physiology, and some of the studies that are going on. So everyone wants to have a happy gut, of course. So what I recommend people do is to be a GI sleuth. The reason I'm here talking to you, and I think it's important, is to understand the complexities of the systems. Understand that our cephalogastric response, the way that we think about our foods, the way we approach our foods actually affects our digestion. So if you're driving into a Sonic and gobbling down a hamburger, you're not giving your body time to make your salivary amylases, make your hydrochloric acid. I recommend that people, to optimize their gut health, really create a ceremony around eating. So prepare the food, go to the store, meet with your families. If possible, light a candle, join with families and friends. I really think that that's how the human being is meant to approach a meal, not gobbling it down through the drive-thru. And also appreciate the interconnectedness of systems. I mentioned mind and stomach acid, vagus nerve and digestion, stress and elimination. These are all things that we can kind of take a look at and say, what causes me my belly pain? Is it because I got to go in and meet with my boss and my stomach starts hurting and I get diarrhea? Maybe there's an association. Again, the only person who can really look at that is you. So at-home testing. What's your salivary pH? The salivary amylase works best at a pH of 7.4. I'm quite certain that upstairs at Nutrition World you can get some pH test strips and find out what your pH is. All right. Gastric acid secretion. I recommend people do what's called a betaine test. These are at-home kits. They don't require an expensive gut test. They don't require a lactulose and mannitol test. These are things you can do in the privacy of your own home. Betaine hydrochloride is a really inexpensive supplement, and what it does is it effectively mimics the presence of hydrochloric acid. So if at the onset of your meal, if you take one betaine hydrochloride, they're typically 600 milligrams, and you get a burning in your stomach and you kind of feel uncomfortable, that's a pretty good information that you probably have enough gastric acid in your stomach. But if you take 1, 2, 3, 4, maybe 5 of these betaines, and you don't sense that there's any burning in your belly, you might have a case of what's called hypochlorhydria, which is low stomach acid. And at that point, I'd recommend that you probably start to supplement with some betaine to improve your digestion. I typically don't recommend people go over maybe 4 or 5 betaines. I think that's sort of getting up there. Beat transit time. We talked about that. Ask yourself the question in the privacy of your bathroom: do your stools float or sink? If all your stools are floating, you might have a higher amount of undigested fats and unemulsified fats that are causing the stool to float. Again, think of the olive oil floating on the water. And of course, if you see any evidence of undigested foods. So in-office testing, I like using something called the Genova GI Effects. Genova is one of the best labs in the country, and this test looks at digestion, absorption, inflammation. It looks at 20 or 30 of the common phyla of bacteria and lets us know, do you have diversity in your gut, or is there not a lot of diversity? We talked about the Genova gut permeability test, hydrogen and methane breath test for bacterial overgrowth of the small intestine. That's another easily done test. What it is you drink lactulose, which is a sugar. If we have upper gastrointestinal bacterial species and overgrowth in our gut, we make a certain amount of methane, and that's breathed out into a container. So we drink the solution, and at 20 minutes, 30 minutes, 45 minutes, an hour, 2 hours, we just breathe into a little container and send it off to the lab and see if there's a hydrogen and methane that's been produced. And food sensitivity testing is important as well. And I didn't spend a lot of time going into this, but I think that foods are one of these substances that is really influencing these dendritic cells. So if there's foods to which we are sensitive, to which we have a delayed hypersensitivity, commonly these are foods that people always eat at the same time. Now the foremost antigenic foods are corn, wheat, soy, and what's the last one? Corn, dairy, thank you. So, but it's surprisingly, you know, I often see people that say, I'm on a dairy-free, soy-free, wheat-free diet. I'm doing everything I can and my gut still hurts, but I have an avocado every morning. And we do gut testing, guess what they're sensitive to? Avocado. So treatment, 4R treatments. This is one of the few times that I use allopathic medicines, is in treating people's gut conditions. So there's a medicine called rifaximin. It's a non-digestible antibiotic for bacterial overgrowth of the small intestine, and it's actually FDA approved for that, believe it or not. I'll use antifungals and antiparasitics. Depending on the individual, I can also use herbs as well. So berberine is a plant alkaloid that's been really well studied. It's very powerful against a number of different parasites and yeast species. Oil of thyme and oregano. These both have some pretty good clinical data behind them looking at treating yeast. Caprylic acid is another great antifungal approach as well. Black walnut, wormwood, oregano, these have been used for centuries in antiparasitic treatments, and they're actually showing a great amount of benefit. And these are just a couple, is Ed in here? Let's just see. These are a couple products that I really particularly like. Ed, you ever heard of biocidin? Do you guys have it? This is great. So one of the ways that bacteria grow in our body is through creating what's called a biofilm, and they create kind of like this sticky, mucousy goop, and they hide in it, which makes them, they can't be detected by the immune system, they can't be accessed by different drugs, and this is a great product that I think can be used to help to break up this biofilm, and there's lots of clinical data behind it. How about Para-X? You heard of that one? All right, here's another one. This is actually, do you stock this? It's only available in Europe, but if you can order it over the internet, and it's sort of a really nice combination of all these herbals. They've got a very detailed, I guess, instruction packet of how to use it, and it's very effective. The next step of the 4R program is to remove. So be sure to remove immunogenic foods, foods that are causing a food allergy. Of course, toxins, pesticides, plastics. This is important to help to improve your immune function. Repair. You know, so often people just reach for glutamine and zinc carnosine. These are a couple of substances that have been really well studied in helping to repair a leaky gut. And I've got a couple of nice papers that look at humans and animal models where a leaky gut is invoked, it's caused, it's demonstrated by the leaky gut test, and then it's repaired by using glutamine and zinc carnosine. So glutamine is a couple grams a day. Carnosine typically comes in about 75 milligrams daily. This is one thing that I think that gets overlooked. The epithelial cells, the cells that line the gut, are regenerating every 2 to 3 days. So it's really important that we've got appropriate B vitamins for the DNA activation, that we have appropriate amino acids, that they have the substrates, the nutrition by which to grow. And I think that one of the things that's associated with if somebody has a leaky gut or a gut that's not working, it reasons what's reasonable to assume that they don't have good amino acid stores because they're not adequately breaking down the proteins and absorbing these amino acids. Slippery elm, marshmallow, diglycerized licorice, and aloe. These are all some nice ideas for symptomatic relief, and those can be taken in gels, in teas, any number of different possibilities. Treatment, I mentioned the betaine challenge. Pancreatic enzymes are also useful if someone's deficient in pancreatic enzymes. Probiotics, and I guess we'll have an opportunity to ask questions about probiotics here at the end of the talk. And the last thing that I think is really important is appropriate amounts of fiber. The cells of the large intestine are dependent on a 4-carbon acid that's called butyric acid. It's their preferred food, and it's really a breakdown product of fiber. Humans do not have the enzymes to break down fiber. The bacteria that live in our gut do, and we can actually measure butyric acid. So, you guys survived. That's the end of my talk. Thank you so much for your patience and for letting me teach you today, and hopefully everyone learned something.